So we're in the foyer of the Sheraton Hotel, and something new that we're going to try. I've caught up with Glenn Brock, who's at the, well, you can see it for yourself because we've actually got the visuals this time. So Glenn, could you take us through Tommotiids?
Sure thing. Thanks, Dave. Yeah, thanks for inviting me. Tommotiids are a, relatively little studied group. They're a group that are unusual in many respects, but extremely important in others. They're unusual in that they produce a skeleton of organophosphatic material, shell material, rather than calcium carbonate, which is the dominant type of biomineralized material in the early Cambrian.
So things like bivalves and gastropods.
Brachiopods and things like that. And interestingly, the Tommotiids are kind of the archetypical or exemplary group of small shelly fossils, and that kind of term, which is widely used now throughout the geological column, really was coined for these early biomineralized bits and pieces of spines and cap-shaped cones and the such. And the Tommotiids are very much a clear small shelly fossil because we, until recently, have had very little understanding of the relationships, what groups of animals these shelly bits and pieces, just like jigsaw puzzle pieces, that fall out of our acid residues. And in the last 10-15 years, myself and my co-authors, which you can see here, some of which are colleagues and some of which are PhD students, have been looking at different groups of Tommotiids from our very extensive sections in the Araui Basin, the Flinders Ranges in South Australia.
Okay, so you say that they fall out of your acid preps.
Yes.
And is that just because of the chemistry? So things like that have the calcium carbonate skeletons, shells that we talked about, gastropods, bivalves, they would actually be dissolved.
Yeah, absolutely. So the processing technique here is we're actually targeting these guys because of their phosphatic shell chemistry. They're resilient to acid treatment, whereas yes, the calcareous shells, which are still there, we have to get them out in more traditional kind of crack out, drill types of methods. We can only use acid treatment for those if they've been replaced by silica or some such replacing mineral. So we have been focusing really on the phosphatica to a great degree, and that's mainly because they're so abundant and they're so diverse, and yet they've been so enigmatic at the same time. They're here at the earliest phases of the Cambrian as well, so some of the earliest biomineralizers belong to this group in the Tommotiids. There's only a few calcium groups that occur before them in the record. And so they must represent, therefore, bits and pieces of skeleton from animals that were there at the very beginning of the explosion.
Okay, so you have some new insights into this group.
We do, and if I can just go on, just to put you into some sort of context, this is a great diagram that Charles Marshall produced for his review on the Cambrian explosion in 2006, and it shows the typical explosion in terms of disparity, body form, and the diversity. And these are all the clades that we know about, from trilobites to mollusks to brachiopods and so on. But you can see out here on the right this little green triangle of small shelly fossils, which Marshall didn't even kind of place within this radiation, and that's because they're just so enigmatic at the time. Even in 2006, many of these things, and the Tommotiids would be amongst them, they couldn't be placed easily into any of the known phyla or clades. And that's because people had not really looked at them. And so they were shoved out to the side in this diagram, and many others as well, because they were thought to be a little bit intractable in terms of getting information out of them. But in the last 10 years or so, it's clear that this group of small shelly fossils, which is really just a descriptive term for, these diverse, mostly tiny, 1 to 2 millimeter, tube, spine, cone-shaped skeletons that come out of our acid baths in absolute huge diversity and numbers. And so we decided that with my students and myself, we would like to have a look at these in much more detail because they're so prolific in the South Australian sections. some examples. This is what we get out of our acid baths. It's just an absolute prolific. I mean, it's almost like just a shell hash. Some of these are beautifully preserved. Some of them are in internal molds, but the bottom diagram shows a whole range of different skeletal elements. Many of them are the Tommotiids, which we're that I was focusing on for the conference today.
Do you have many students picking through all this stuff?
Yes, I pay quite a few undergraduate students. That's good. They get some experience about acid leaching techniques. So we put them through the whole process from getting the rock from the field, which needs to be kind of cleaned down, get rid of all the lichens and modern day things on it, and then they're weighed, and we put it through mostly acetic acid, which is dilute to 10%, and we leave the sample in for 4 to 5 days mostly, and that etches enough of the surface to release the insoluble phosphatic or sometimes phosphatized internal moles, which we can see in this image here. This is where we get it from. This is some of the images here of the Wilca-Willina limestone, and the image on the top right is the Mernmerna formation. These are just bristling with these tiny sclerites. You can see them in hand specimen in the field, but they really only become manifest after the acid treatment. Where they are there, but just about every sample you take, fist size or potato size sample, will have some form of shelly material in them. It's very rare that we have a barren interval. And on the left-hand side, you can see it's just a summary of the stratigraphic successions that occur in the Flinders Ranges for the Lower Cambrian, and the lowermost dotted line indicates where we begin to get really diverse, the first occurrences of small shelly fossils. And we get them throughout the entire succession of about 5 kilometres of Lower Cambrian material. So there's lots of changes in the diversity and the types of small shelly fossils that occur through this period of time.
So is it just the Flinders Ranges that you get these, or is it a worldwide?
Yes, they're worldwide. The Tommotiids are perhaps the most diverse in Gondwana, in East Gondwana. So
that's the lower hemisphere?
Yes, so that's the lower hemisphere, which includes, of course, East Gondwana includes Australia and Antarctica in particular. But they occur throughout Siberia, Mongolia, the UK, Laurentia. All of these places also have small shelly fossils. Interestingly, some provide a means to correlate across these continents, but often many of these big paleocontinents have their own collection of small shelly fossils that are unique to these particular areas. So high degree of endemism, as we would call it, so that they're restricted in terms of their distribution, and that's what we find in South Australia as well. But the Tommotiids themselves, as I mentioned, they're the exemplar small shelly fossil. Their key features are they have this unusually an organophosphatic composition. There are very few organisms that have a phosphatic composition through geological time. There are a group of brachiopods which have phosphatic. There are conodonts have some phosphatic material, and indeed our bones, of course, are calcium phosphate. But it's not used all that much in the geological record for biomineralization. But the Tommotiids, very early on, seem to have flicked the switch for phosphatic biomineralization. And it's really interesting because they're quite variable. They're pretty sloppy in the way that they build their skeleton. Some of these images here look absolutely beautiful, and they are, but there's always lots of disruptions and kind of pathologies in the shell that indicate that these were animals that were just kind of getting going with the biomineralization, and we can see lots of kind of anomalies in the way that the shell is laid down. The important thing about them is that they form by what we call marginal accretion, which just means that they lay down a bit of shell around the edge of the organism, probably where a soft mantle was, very much the way that brachiopods and bivalves lay down their skeleton today.
So if we looked at the top right, the Macquarie,
Yep.
so the oldest bit of the fossil is right at the peak,
Yep.
and then it just makes the concentric rings
and
around
gets
the outside.
larger and larger and larger. That's absolutely right. And the same with this coniform one, Lapuathella, over here on the left. Its oldest part is right at the tip, and then you can see the growth lines where we've accreted and grown this long conical shape. Some of them become kind of coiled or torted or twisted in their development, but they all grow in pretty much the same way.
Right.
The interesting thing is, what we do find is each one of these little skeletal elements is called a sclerite. Now, what's clear is we can see that in the Tommotiids there are 2 to 3 distinct sclerite types, which we can see by their same ornament, same internal structure, that we can kind of see that they belong probably together, indicating that the animal had more than one sclerite. There was not one single animal living inside this. It was part of a bigger composite skeleton which is made up of these sclerites, sometimes 2 to 3, and we call this composite skeleton a sclerotome. And how the big mystery has been in what shape or form the sclerotome was amongst the Tommotiids, and up until recently we had no real idea. We just found the jigsaw pieces, but they were never fused together into the complete skeleton. However, our work and the work of my students and colleagues, we've been able to kind of work out at least what some of them look like. And some examples are shown here. These are called Eccentrothecamorphs, and they're a group that's named after the key taxon, which is called Eccentrotheca, and it's called Eccentrotheca because it has an eccentric way of laying down its skeleton. It's quite variable, but essentially there are 2 types of sclerites, and you can see it mostly, or probably best, in the upper left-hand image. It's a reconstruction of a tube, and we know that because we've got these beautiful specimens below of the actual sclerotome. So we know how these sclerites, towards the base, how they fused together. They were kind of flattened, fused, and open sclerites, which gave way up the tube to more flattened, compressed triangular sclerites. And we'd always recognise these as Eccentrotheca sclerites, but we never knew how they went together. But we sampled particular levels in the Wilkawillina limestone, and we were lucky enough to retrieve the complete sclerotome in 3 dimensions. And so we now know that...
Oh dear, someone's making smoothies.
Hope there's no sclerites in those.
Yeah.
And so we can see that it's a tube-like skeleton, which was kind of unexpected. People were thinking it was going to be like a slug-like mobile animal with a kind of a back full of sclerites kind of arrayed over and it was going to be moving around. But so it was somewhat of a surprise to us, but a delight to find that they were seemingly fixed, sessile, and probably filter-feeding tube-like organisms.
Yeah, I remember the interview we did with Jakob Winter on Paleozoic Problematica, where we had multiplicophorans,
yes,
which were organisms that had kind of like chitons that had these little sclerites.
Sclerites all over the surface, and these certainly were kind of mobile, epibenthic, probably grazing or deposit-feeding forms. Same with the Holkeriids, the famous Holkeriids, which have a similar design. But these guys were seemingly much simpler, but yet the skeleton, made up of all of these tiny sclerites, fused together to form this protective conical skeleton, which we think would have attached to kind of like hard substrates. Like in the Cambrian we have our first reefs. So there were archaeocyathids, these cup-shaped calcitic sponge-like group, and these form the first reef mounds, and these guys lived in and amongst these reef mounds in crypts and cavities, and we see this very clearly in the South Australian material. The interesting thing about them, when we look at the ultrastructure, is that they have a lot of things in common with brachiopods. I mentioned one of the groups of brachiopods that is called the linguliform brachiopods. They also produce an organophosphatic composition. So there's relationship number one. They've got a similar type of relationship. The difference is that the Tommotiids, of course, are either tube-like or they do, in Micrina, for instance, come down to just a pair of valves that join together. And when we look at the ultrastructure, we see various tube-like penetrations that go through the shell, and these are called setal canals, and this is where soft sensory setae would have emerged from the shell and probably sensed the environment. We find the same structures in true brachiopods, early Cambrian brachiopods. Here's an example called Cetatella, and they have the same type of canals for setal insertion. So this provides us with a link. Not only are they organophosphatic, they have the same sorts of structures in terms of size, shape and morphology in the shell, which indicate that these Tommotiids probably have some sort of relationship to brachiopods. Here's another example. This is Micrina again down in the bottom left, and it has a clear larval shell. So in other words, it probably had a planktonic larvae that it would have dispersed with and settled and then formed into the fixed sessile adult. And if we look at the shell a bit more, we can see that there's also, you can see down in the bottom right, one of these, it's labelled TU, it's one of these setal canals, but they also have these columns that run perpendicular through the phosphatic laminae. And if we look at Cetatella, which is a true lingulate brachiopod, we see the same thing. We've got these columns that run through the horizontal organophosphatic sheets and very similar in structure. So it's kind of indicating to us that these, at least the Eccentrothecamorph Tommotiids, probably have a relationship to the phosphatic brachiopods. Another example, and this is really cool, this is in the top row we've got a Tommotiid called Patomitra, and it has a really distinctive reticulate ornament on the surface when you put it under the SEM. Below is another type of phosphatic brachiopod called the Patorinids, represented by a taxon called Ascopasma, and you can see that it has a remarkably similar type of ornament, this reticulate, very beautiful external ornament. You can only really see it under the microscope, you can't see it in the field. But interestingly, if we cut through the shell, we can see that this is just not surface, it actually pervades right through the shell, this reticulate structure, which is quite unique. When we first found it in this Tommotiid called Patomitra, and when we did the same thin section through Ascopasma, lo and behold, we found the same unique shell structure. It's not found in any other organophosphatic groups. Again, linking the Patomitrids with the brachiopods, the Tommotiids with the brachiopods. So it seems fairly clear now that at least this group of Tommotiids has found a home somewhere in the ancestry of the evolution of the brachiopods. This group, however, of Tommotiids is still a big mystery. And my talk today kind of focused on these guys a little bit more because we know little about them. We call them the Caymanellans after a genus called Caymanella, which is shown down here in the bottom left. They're beautiful, sometimes they're coiled sclerites. The scale bar here, I think, should be around about 500 microns or half a millimetre. So they're not too bad. Some of them are 200 microns, you can see here. But their key features are they tend to be more conical or coniform in shape, but they have this very distinctive radial folds and plicae which run from the tip down, kind of like folds of a curtain around the edge of the sclerite. At the same time, they've got a concentric ornament, like all Tommotiids, because these are representing the accretionary growth of how they were formed. But the interesting thing about these guys is that we've found no fused elements ever. Well, one example. So these are like a jigsaw puzzle in that we can, for instance, find different sclerites. If you look at Dahliadia ajax here, there's a symmetrical sclerite below the label, and then there's an asymmetrical twisted sclerite above. And they have the same kind of ornament and shape and so on. So we think they go together, but in what type of sclerotome they had remains a big mystery. And what we've been doing is trying, and we have now tens of thousands of these guys. They're just amazing diversity and numbers. But the thing that they do have is this also this amazing reticulate ornament, like the Eccentrothecamorphs, except this is just at the surface. It doesn't pervade the shell structure whatsoever. So there must be some slight difference here. So they're similar in that they have this beautiful reticulate. You can see here this close-up showing this kind of band of smooth area, quite thin, followed by this thickened reticulate area, and then are repeated. And so this is this marginal accretion, how it's growing in these bands of phosphate and laying it down with this wonderful ornament. Now, you might ask me what that may mean functionally or embryologically, and we're not quite sure yet. So we're still thinking about whether this means that these erupt through soft tissue. So like your teeth grow through the gum and leave behind kind of telltale signs. This may be the case with these guys, but we're not sure because we haven't, of course, found them in association with soft tissue. Unfortunately, there's no Tommotiids found in any of the Lagerstätten, these deposits where we have preservation of soft tissue, soft material. They're always found as shelly material only.
Why is that if Tommotiids are really common throughout all of the early Cambrian?
I think it's mainly because they prefer shallow water tropical environments in the main. You can find them outside of that, and of course most Lagerstätten are found in slightly deeper water kind of vent beds, and so these are much more carbonate based organisms, whereas the Lagerstätten tend to be in kind of muds, shales, and so on. So, and the Tommotiids just seemingly didn't prefer this type of environment. Now, in my talk, I just use Daleatia as an example, mainly because it's so diverse in South Australia. We've got thousands and thousands of sclerites, and we can match the morphology up of the sclerites quite nicely. And we can recognise different species in Australia and Antarctica. And this is the only place, somewhat frustratingly, that Daleatia occurs. It made Australia its home in the early Cambrian, but obviously did not have particularly good powers of distribution, and so it's found only in Australia and Antarctica, and of course Antarctica was juxtaposed. It was sutured to to Australia at this particular time. And interestingly, when we look at Daleatia, we can see that there are a number of different sclerites of the body, and you can see I've called them A1, A2, B, C1, C2. So there's 5 different sclerite types that we can clearly associate it with this particular species, Daleatia macroptera. The A sclerites are symmetrical down the median plane, so they're bilaterally symmetrical, yet the B sclerites and the C sclerites occur in left and right twisted forms. So this is very unusual. We don't see this in the Eccentrothecamorphs. These guys, because they form a tube, don't have these asymmetrical twists. And this kind of gives us an indication that these guys might have a different sclerotome design than the Eccentrothecamorphs, because there's more sclerite types, and it's very hard to put these into a kind of a tubular model. Would be lovely to find a complete or partially complete one, but at this stage we don't. But the hints are there that this might be a more complex sclerotome. If we look at sclerite ratios, I won't dwell on this, we get a similar picture in that the C sclerites, these kind of flattened ones that are twisted, are the most diverse, indicating and they occur, the most numerous, and they occur in this left and right form, and we think that they probably occurred on the left and right sides of the body based on this. And of course the A1s and the A2s are symmetrical, and they probably occurred along the median plane of the body. So you might see where I'm going with this now.
Yep.
We think, and when we look at the ultrastructure of Daleatia, it's completely different. You can see in this slide here, it's got very finely compacted laminae of phosphatic material, completely different to what we see in the Eccentrothecamorphs with this organic network of reticulate material, or with canals running through it. Nothing like that in the Caniculate. It seems it's just a massive shell structure built up by subsequent very tightly packed organic layers. They also, interestingly, the A sclerite always has 2 openings on the tip, and the C sclerite has 1 opening. Now, we assume that there's some sort of sensory, cetal, but we don't know. There's never been anything found. But that's very consistent throughout. So when we kind of put all of this together with one specimen is always, n equals 1 is not really great, but it's one specimen where we have an A1 sclerite, you can see down the bottom left here, with a C1 sclerite attached to it. And you can see if the A1 sclerite is in the median plane of the animal, the C1 is on the left-hand edge of the sclerite, and since they occur in left and right pairs, it's probable that there was another C sclerite on the right-hand side. And if we start to build the animal with CT scans of the sclerites, we can see that they actually do fit together rather nicely. So that you can see in these reproductions here, the colours of course are artificial, you can see an A sclerite here with the C sclerites attached quite nicely based on the shape of the available space. And so we're coming to a conclusion that we think that Daleatia and probably all of the Caminellans had a much different type of sclerotome than the Eccentrothecamorphs, suggesting indeed that they may not actually be related. They might have a different ancestry, and so that the Tommotiidae may in fact be what we call paraphyletic. It's got completely different signals for clades within that one group. And this is a reconstruction on the right-hand side, done by one of my students, Marissa Betts.
I recognise that from the Paleocast art competition.
Indeed, which she did submit. I think this is slightly updated because myself and my co-author, Christian Skovsted, at the Natural History Museum in Stockholm, we had a few issues with her first interpretation about the numbers of sclerites, but Marissa's now modified it beautifully. This gives us an idea of what we think the sclerotome might have been like, and the numbers are based on the proportions of the sclerites that we find based on the tens of thousands that we get throughout the record. But it's not, we weren't the first to come up with this idea. You can see over here on the left that Evans and Rowell in 1990 also came up with a similar sort of design for Daleatia, but I think that doesn't take into account the variation and the number of sclerites quite as well as our new model does. But it does show that the big thing that comes out of this is that these Tommotiids and these small Shelly fossils still have a lot to give in terms of understanding the relationships of these early clades. So if we look at this phylogenetic relationships here, and this is just a indicative diagram, it's not a cladistic analysis, because it's hard to do that when we don't have all of the sclerotome material available. So we're being a bit cautious here, but we can see some trends in that the Eccentrothecamorphs certainly belong probably in the ancestry or the stem of the brachiopod, the organophosphatic brachiopod groups. There's the linguliform brachiopods and the paterinid brachiopods. They're the two main groups, and we think we can see that signal quite nicely there. The Caminellans, however, over here in blue, these are kind of an offshoot, a sister group, if you like, probably related because they've got an organophosphatic composition and they produce sclerites. There's similarities in the outer surface reticulate structure, but they do have a different way of laying down their phosphate, and we think now that they have a different sclerotome as well. What they're related to, you could put a big question mark up the top there, and we just don't know at this stage. But it's cool. We're starting to make some real strides in putting these small Shelly fossil groups into kind of the tree of life. And this also fits in with the first appearances of Tommotiids. You can see in this diagram here, the first appearance of Tommotiids is around about 536 million years ago, quite low down in the Fortunian or early part of the early Cambrian. And you can see in the red box just below that, the incoming of the phosphatic and calcareous brachiopods come in a bit later, making it plausible that at least for the Eccentrothecamorphs, they could be, the ancestors of the true phosphatic brachiopods. So it could well be that in fact the Tommotiids were at least the Eccentrothecamorphs. The bottom part of that could be moved down and they could be linked to the phosphatic brachiopods. So that's going to be probably one of the key outcomes of our work. And so, I mean, the thing that I wanted to get out of this is that Shelly fossils, in many cases, are no longer as enigmatic as they once were, and the more work we do on them, the more clear it is that we can place them at least within the major phyla, brachiopods, mollusks, arthropods, and many of these previously enigmatic forms now are starting to find homes, and the Tommotiids is one of these. At least with the Eccentrothecamorph Tommotiids, we think they're clearly related to the brachiopods, both in terms of the composition, structure, ultrastructure, form, and so on. Whereas the Caniculate, well, we're not quite sure. They may have had a sclerotome like Halkieria or these multiplicophorans that we talked about earlier. That's our favoured view. But if we find a big tube next week when we go next to the field, then we'll have to rethink completely this. So that's quite speculatory about the Cambrian sclerotome, and it's based on relatively limited evidence. But I think it does show that these sclerites are really important in kind of working out character traits early on in the evolution of animals.
That's great then, Glenn. I guess send your questions in.
Yep, please do. Happy to answer.
All right, thank you very much.
No worries.